Hi everybody. Fusarium polycephalum, also referred as slime molds, belong to the supergroup Amebozoa, phylum Mycetozoa, and class Mixogastria. P. polycephalum, often referred to as the many-headed slime, is a slime mold that inhabits shady, cool, moist areas such as decaying leaves or logs. It is sensitive to light. In particular, light can repel the slime mold and can be a factor in triggering spore growth. Characteristics: this protist may be seen without a microscope. P. polycephalum is typically yellow in color and it eats fungal spores, bacteria, and other microbes. P. polycephalum is one of the easiest eukaryotic microbes to grow in culture and has been used as a model organism for many studies involving amoeboid movement and cell motility. CBL001 Phoma sp. Pleospora is a synthetic model organism, and that is also called silent superbug or Morgellons. Now what's very interesting here is the fact that light can repel the slime mold. So if we want to repel the slime mold, we need to be in light. The life cycle, the main vegetative phase of P. polycephalum, is the plasmodium, the active streaming form of slime molds. The plasmodium consists of networks of protoplasmic veins and many nuclei. It is during this stage that the organism searches for food. The plasmodium surrounds its food and secretes enzymes to digest it. Oh, I tell you, trying to make a video when you live on a highway next to an auto body shop is getting pretty hard to do. But just bear with me because this video is definitely worth listening to. Now, what is interesting here is the fact that this plasmodium consists of a network of veins, and Morgellons patients have reported that they have a network of veins running through their flesh. Now also what's interesting here is the plasmodium surrounds its food and secretes enzymes to digest it. If environmental conditions cause the plasmodium to desiccate during feeding or migration, Fusarium will form a sclerotium. The sclerotium is basically hardened, multinucleated tissue that serves as a dormant stage, protecting Fusarium for long periods of time. Once favorable conditions resume, the plasmodium reappears to continue its quest for food. As the food supply runs out, the plasmodium stops feeding and begins its reproductive phase. Stalks of sporangia form from the plasmodium. It is within these structures that meiosis occurs and spores are formed. Sporangia are usually formed in the open so that the spores they release will be spread by current winds. Spores can remain dormant for years if need be. However, when environmental conditions are favorable for growth, the spores germinate and release either flagellated or amoeboid swarm cells, motile stage. The swarm cells then fuse together to form a new plasmodium. Now let's just go over this a little bit. The yellow color, Morgellons patients report an amber goo that they find in their bodies. Also the fact that it eats fungal spores and bacteria and other microbes could explain why Morgellons patients or silent superbug patients rarely get sick with any bacterial infections. They can basically do their own biopsies and never get infections, and that's a fact. The life cycle. Now, what's interesting about this is the fact that it consists of the network of veins, also that it is searching for food, and that the plasmodium secretes enzymes. So what effect does that have on our bodies? Also, it's speaking of the right environmental conditions in order to cause the creation of something called a sclerotium, which is basically hardened multinucleated tissue. Morgellons patients have very, very hard matter within them that is trying to come out of the body, and also the CBL001 Phoma sp pleospora or silent superbug videos spoke about multinucleated. Also, those videos and research and proof spoke of a dormant stage, and every Morgellons patient knows that there are very certainly different stages to the process. So that would explain the dormant stage. You know that many insects also have a dormant stage. They can lie dormant for many, many years and then come back to life later on. It talks about the reproductive phases and the stalks of sporangia, and also how reproduction occurs through meiosis, where the spores are formed. Okay, streaming behavior. The movement of P. polycephalum is termed shuttle streaming, and that is characterized by the rhythmic back and forth flow of the protoplasm. The time interval is approximately 2 minutes. The forces of the streaming vary for each type of microplasmodium. The force in amoeboid microplasmodia is generated by contraction and relaxation of a membranous layer, probably consisting of actin. And I know one of the researchers talked about actin. And actin is a type of filament associated with contraction. The filament layer creates a pressure gradient over which the protoplasm flows within limits of the cell periphery. The force behind streaming in the dumbbell-shaped microplasmodia is generated by volume changes in both the periphery of the cell and in the invagination system of the cell membrane. Okay, now what is interesting is the idea of contraction and relaxation. Also, it talks about the filaments. Very, very interesting. Situational behavior. Fusarium polycephalum has been shown to exhibit intelligent characteristics similar to those seen in single-celled creatures and eusocial insects. So it has Intelligence! And I've been telling people for a long time that these fibers move of their own intent. Maze solving. A team of Japanese and Hungarian researchers claim that a specimen of P. polycephalum was able to navigate a maze made of agar, using the shortest route possible when two pieces of food were placed at two separate exits of the maze. And I'm sure you've probably seen the videos on this slime mold going through the maze. There are many YouTube videos on it. Event anticipation. By repeatedly making the test environment of a specimen of P. polycephalum cold and dry for 60-minute intervals, Hokkaido University biophysicists discovered that the slime mold appears to anticipate the pattern by reacting to the conditions when they did not repeat the conditions for the next interval. Upon repeating the conditions, it would react to expect these 60-minute intervals, as well as testing with 30 and 90-minute intervals. So the slime mold has the ability to actually anticipate events. Nutrient regulation. P. polycephalum have also been shown to dynamically reallocate to apparently maintain constant levels of different nutrients simultaneously. In particular, specimen placed at the center of a petri dish spatially reallocated over combinations of food sources that each had different protein-carbohydrate ratios. After 60 hours, the slime mold area over each food source was measured. For each specimen, the results were consistent with the hypothesis that the amoeba would balance the total protein and carbohydrate intake to reach particular levels that were invariant to the actual ratios presented to the slime mold. Simulation of road networks. With more than two sources, the amoeba also produces efficient networks. In particular, the pattern connecting multiple food sources was shown to form efficient network structures like cycles and Steiner minimum trees. In a 2010 paper, oat flakes were dispersed to represent Tokyo and 36 surrounding towns. P. polycephalum created a network similar to the existing train system and with comparable efficiency, fault tolerance, and cost. Similar results have been shown based on road networks in the UK and the Iberian Peninsula, Spain and Portugal. Oh, listen to this. Integration with electronics. The organism's reaction to its environment has also been used in a USB sensor and to control a robot. Okay, no doubt in my mind, the purpose of putting this slime mold into us, to integrate it with us, is to do exactly that. Enable them to put a USB sensor into us and to control us like robots. Please share this video. Please share it wide and far. Computing. In a book and several preprints that have not been scientifically peer-reviewed, it has been claimed that because Plasmodia appear to react in a consistent way to stimuli, they are an ideal substrate for future and emerging biocomputing devices. They are ideal substrate for future and emerging biocomputing devices. It has been reported that plasmodia can be made to form logic gates. Logic gates, in particular, plasmodia placed at entrances to special geometrically shaped mazes would emerge at exits of the maze that were consistent with truth tables for certain primitive logic connectives. However, in the preprint, when these primitive gates were connected to form higher logic functions, the plasmodium ceased to produce results consistent with the expected truth tables. Consequently, the composed gates were validated instead using a simulation speculated to model the streaming processes within a plasmodium. An outline has been presented showing how it may be possible to precisely point, steer, and cleave plasmodium using light and food sources. So light and food sources are very important to investigate further. Very important to check into these other articles, which are the inline references. The first one refers to intelligence maze solving by an amoeboid organism. 2. Amoebae anticipate periodic events. Top 100 stories of 2008. Slime molds show surprising degree of intelligence. 4. Amoeboid organism solves complex nutritional challenges. 5. Brainless behavior. My exomycet chooses a balanced diet. Number 6. Obtaining multiple separate food sources. Behavioral intelligence in Physarum plasmodium. 7. Rules for biologically inspired adaptive network design. 8. Brainless slime mold builds a replica Tokyo subway. 9. Road planning with slime mold. If Physarum built motorways, it would route M6, M74 through Newcastle. International Journal of Bifurcation and Chaos 20. 10. Rebuilding Iberian motorways with slime molds. 11. Biosensor puts slime mold at its heart. 12. Robot moved by slime mold's fears. So the robot can be moved by the fears that the slime mold has. So the slime mold actually has fear. Can you believe that? 13. Physarum machines. Computers from slime mold. 14. Slime mold logical gates exploring ballistic approach. And 15. Steering plasmodium with light. Dynamical programming of Physarum machine. Now we have to realize that there was an article or a book written by Barbara Marciniak way back, at least 25 years ago, talked about the Pleiadians and a channeled message and about how all of us humans were going to be changed, our bodies, and we were going to be changed by light encoded filaments. And those light-encoded filaments were going to change our strands of DNA. This is actually happening. But who's behind it? Is it the Pleiadians or just a bunch of mad scientists? Then there's another article, general references, studies of microplasmodia of Physarum polycephalum. When you look at my YouTube channel, Morgellons cover-up, I'm certain you will see this slime mold in probably all of over the 400 and some videos that I have on there. Please share this information far and wide. Thank you so much for watching this video.